All right. So welcome to our first presentation in the embryology series for this year. My name is NIGZ Gastrulation. I will present on gastrulation and the trilaminar disc, which is basically what's going to be happening in the third week of development. Right. Confirm you can see my screen well and clear.
Yes, we can see it.
All right. So I've outlined my presentation in a way that I'm just going to give a brief background on how we get to the third week of development. Then we're going to talk about gastrulation and then move on to the derivatives of the germ cell layers, right? So these are our learning objectives. By the end of the presentation, one should be able to describe the process of gastrulation and also describe the role or be able to understand the role of the primitive streak in gastrulation, then also describe some basic molecular regulation with regards to gastrulation and the fate of the germ cell layers. Then lastly, we'll then describe the fate of the germ cell layers in terms of what they do form, mainly focusing on the ectoderm. Then, of course, we'll also do mesoderm and endoderm. Right, so I'll start with a bit of background information. So initially, what happens during development is you're going to have gametogenesis, that is going to be oogenesis and spermatogenesis for the male and the female respectively. And then you're also then going to have fertilization, that is after you've had completed your oogenesis and spermatogenesis. So having done that, you're going to start the process of cleavage, where your cells are going to start dividing and increasing in number. So what you have will be a zygote, which is not increasing in size, but is simply increasing in cell number due to the mitosis that will be happening. So because you'll be still be having a zona pellucida outside, you're not going to be able to increase in size, but you're going to increase in cell number due to the mitosis and the cleavage that will be happening. Then you're also then going to have formation of blastocyst, which then loses the zona pellucida. Then it undergoes the process of implantation. And remember, implantation will be happening either on the anterior wall or the posterior wall of the uterus. Then after you've had implantation, remember that blastocyst will be having an inner cell mass and an outer cell mass. Outer cell mass is the one that you then say in the second week of development will form your syncytial trophoblast and the cytotrophoblast. Then the inner cell mass will give rise to the epiblast and the hypoblast, right? So with that sort of short background, when you now come to the third week of development, the most characteristic event that will be occurring in the third week of development is going to be gastrulation. And this is basically the beginning of morphogenesis. So by definition, gastrulation is the process that establishes all three germ cell layers. That is going to be endoderm, mesoderm, and ectoderm. That is from inside going outward. And in that process, we form what we call a gastrula. So you can call it a gastrula when it's now a trilaminar disc, right? So to start the process of gastrulation, there's going to be formation of the primitive streak. So the primitive streak forms on the cell surface of the epiblast. Remember, we said inner cell mass was going to divide into epiblast and hypoblast. This happens in the previous week, which is going to be the second week. So in this third week, you're going to find that on the cell surface of the primitive streak, you're going to have, of the epiblast rather, you're going to have formation of the primitive streak, right? And this primitive streak forms in such a way that you're going to have thickening of cells towards the center. Then they start to elongate going to the posterior end. Once you form the primitive streak, cells of the epiblast, which is the same cell layer that is which we are having formation of the primitive streak, cells of the epiblast are going to start to invaginate through that primitive streak. And when they start to invaginate, that's when we are going to start having the process of what? Of gastrulation in terms of formation of endoderm, mesoderm, and ectoderm. We'll get to that shortly. So for now, we have said that we have formation of a primitive streak on the cell surface of the epiblast. And then cells of the epiblast are then going to start invaginating through that primitive streak. So for all this to happen, you're going to need fibroblast growth factor 8, which is a protein that is encoded by a gene which you just call fibroblast growth factor 8 gene, right? So that protein, which is going to be fibroblast growth factor 8, is the one that is responsible for cell migration and specification. So by this, I mean that for these cells to start invaginating through your primitive streak, you're going to need that fibroblast growth factor 8. And also for you to have those cells having specification, for them to say these are going to form endoderm, these are going to become mesoderm, these are going to become ectoderm, you're also going to need that fibroblast growth factor 8 protein, right? So what fibroblast growth factor 8 is going to do is that it's going to downregulate a protein that we call E-cadherin, right? So if you remember from your physiology, or I think the general cell histology, for cells to be adhering to one another, you need some form of adhesive molecules, which include E-cadherin. So cells of the epiblast, they're going to have E-cadherin holding those cells together like an epithelium. So when you now want those cells to start detaching, for them to then invaginate through the primitive streak, you then need to downregulate your E-cadherin, right? And for you to downregulate your E-cadherin, you need fibroblast growth factor 8, right? Then remember we were having an epiblast and a hypoblast, right? So these cells of the epiblast, they're going to start by becoming flask-shaped when you're having downregulation of E-cadherin. They're going to change shape from a columnar shape to a flask, sort of like a shape, and they start to detach from one another. And when they start to detach from one another, you now start to have these cells invaginating through the primitive streak. So I'm going to show a diagram. This is around the beginning of the 3rd week, which is around day 14, 15. So when you look at this diagram here, in blue, that's your original epiblast, right? Then in gold, where the red laser pointer is, that's your original hypoblast. So initially you were having epiblast and hypoblast. So on the cell surface of the epiblast, that's where you form the primitive streak. And this primitive streak, remember we say that it's going to form initially within the central part, then it starts to elongate going caudally. Then the cranial end of your primitive streak, it starts to proliferate. As you can see on this cranial end here, it's like thickened. So it's going to thicken or proliferate, and it forms what you call the primitive node. And associated with the primitive node, there's going to be a depression there, which is your primitive pit. But what we are concerned with for the purpose of this presentation is the central streak here, which is the primitive streak, through which cells of this epiblast are going to invaginate, right? So we have an epiblast, then we have a hypoblast, then we now have the primitive streak. Now, day 15, 16, we're now going to start having invagination of those epiblast cells into the primitive streak. And when they start to invaginate, you're going to form 3 germ cells, right? Which is going to be ectoderm, mesoderm, and endoderm. Ectoderm is the one that is going to be on the outside. Then endoderm is the one that is on the inside. Mesoderm is the one that is in the middle. So the prefix mesos means amongst or in between. So mesoderm is the one that is in between, right? So when you look at this diagram again, you'll notice that the cells of the epiblast that invaginate first and displace the hypoblast, those are going to form the endoderm, which you can see on this picture here. So the cells that displace the hypoblast, they form the endoderm, which on day 16 here, it's appearing in, I think it's gold or yellow. I'm a bit colorblind. Then the cells that come to lie in between, the cells are formed by your endoderm, and those that remain on the cell surface. Those that come in between, which in this picture appears red, that's going to be mesoderm. Remember, mesos, amongst or in between 2 things. Then cells of the epiblast that remain on the cell surface are the ones that are going to become ectoderm. So the basic process of gastrulation is leaving us with 3 germ cell layers, which is going to be ectoderm, mesoderm, and then endoderm, right? So having said that, we now have 3 germ cell layers, which is your ectoderm, mesoderm, and endoderm. So now we want to describe the molecular or a bit of molecular regulation that occurs and determines the fate of both or all the 3 germ cell layers, which is actually your ectoderm, mesoderm, and endoderm. So we have fibroblast growth factor, factors, and bone morphogenic protein 4, right? So there's going to be expression of fibroblast growth factor. Remember I said your primitive streak cells are going to express fibroblast growth factor 8, which I said was responsible for the cell migration and the cell specification. So in this case, what's going to happen is that your fibroblast growth factors rather will repress or will suppress the transcription of bone morphogenic protein 4. And on the other hand, it's going to upregulate the expression of certain proteins, which is codin and nogin, right? So nogin and codin, what they do is they are the ones that regulate the levels of bone morphogenic protein 4. So the reason why we are focusing more on bone morphogenic protein 4 is that the ectoderm, which is the outer layer of our 3 germ cell layers, is the one that is mostly related to formation of neurostructures. So we want to specify the ectoderm into 3 separate regions, which are going to be number 1, the neural plate, which we'll describe shortly with the process of neuralation, which is formation of the neural tube. Then number 2, we want to also have a neural crest cell region. Then number 3, we also want to have remaining ectoderm, which would be the surface ectoderm. Right. So you'd find that bone morphogenic protein 4 is the one that is going to help us with induction of those 3 regions. So to make it easy, if I put the example this way, of the three regions, one region is going to have high levels of bone morphogenic protein 4, or let's just say BMP4. The other region is going to have intermediate levels. Then the other one is going to have very low. So you have high, intermediate, and very low. So in the order that I gave you, the neural crest cells of the region is the one that is going to be central, or the intermediate one. So that's where you have intermediate levels of BMP4. But for all this to happen, the background is that we need noggin and codin. And for us to have noggin and codin that regulate your BMP4, you need to have fibroblast growth factors. And those fibroblast growth factors are being expressed by your primitive Strix cells, where the cells were invaginating, right? Then for the cells in that region to then become neural crest cells, we also need certain proteins, which the main one being snail. And when we then discuss the individual components as to what happens to neural crest cells, you're going to find that these cells are going to migrate. And for them to migrate, we also need a molecule called SLAG. So there is SNEL and SLAG. SNEL is for specification for the cells as neural crest cells. Then SLAG is for the cell migration. Then where you have very low levels, right? That's where you're going to have the neural plate. So the central part, that's where you're going to have very low levels of your BMP4, and that's where you're going to have formation of the neural plate. That's for the ectoderm. Then where you have very high levels of BMP4, this is where you're going to have surface ectoderm. But I also want you to know that the high levels of bone morphogenic protein 4 will also induce part of the mesoderm to form lateral plate mesoderm and intermediate mesoderm. So we're going to realize that mesoderm will be divided into 3 portions. And for 2 of those portions, they're going to be specified by high levels of bone morphogenic protein 4. And that's going to be lateral plate mesoderm and intermediate mesoderm. The low levels, which we said cause specification for the neural plate region, they're also going to induce the other part of mesoderm to become paraaxial mesoderm. So the take-home message is that the BMP4 is not only going to regulate the structures of the ectoderm, but it is also going to regulate what's happening in the mesoderm. Right. Okay. Then, so this is a diagram which is now showing what we were just describing. So what you have on the surface here is ectoderm, and we can see 3 colors. There is the purple part, there is the green part, then there's the blue part. So the purple part here, this is your neural plate, where you remember we said you're going to have low levels of your BMP4. Then in green here, that is going to be where you're going to have your neural crest cells. Then and we had intermediate levels of BMP4. Then in blue, that's where you have high levels of BMP4, and that's where we're going to have surface ectoderm, right? So for the ectoderm, through different levels of BMP4 being expressed in that region of ectoderm, we're going to have neural plate, then we have in green, the neural crest cells, then we have in blue, the surface ectoderm, right? And then once we have had this, we're going to start the process of neurulation. So neurulation is now formation of the neural tube from the neural plate, right? So what simply happens first is there is formation of neural folds on these neural plate borders. So remember the border between the neural plate and the neural crest cell, we're going to have formation of folds or thickenings on those outward ends. And once you form neural folds, they're going to converge towards the center, right? Which is what you're going to describe when we then do development of the central nervous system. That will now be the starting point. So neural folds are then going to come in opposition and they're going to join and they detach from the what? From the cell surface. And when they detach, we come here and form a neural tube. So when this neural tube detaches from the surface ectoderm, can you also notice that in green here, which is your neural crest cells, they also detach from the what? From the cell surface, right? So basically, the molecular regulation is there to help us understand what is going to happen with all your germ cell layers as to how you come about moving from a general ectoderm layer to neural plate, neural crest cells, then the surface ectoderm, then mesoderm. We said we also divide it into three. Sorry, is that a question? All right. Right.
No, it's not a question.
All right. So what we now have are the three germ cell layers and their divisions. So when you look at ectoderm in general, ectoderm is going to form organs and structures that maintain contact with the external environment. I remember back in the day, we used to have a misconception to say that structures that had stratified squamous are the ones that came from ectoderm. It's not entirely true. For example, you can look at the esophagus. So ectoderm is going to form structures that maintain contact with the external environment. For example, skin, right? That's one example. Then when you now want to look at the specifics of the ectoderm, remember we now have three structures. We now have a neural plate, which becomes the neural tube. We now have neural crest cells. We now have surface ectoderm. So the surface ectoderm, what it does is it forms the epidermis of the skin. Remember, skin is going to have two layers, epidermis and the dermis. So the epidermis is the one that is going to come from the surface ectoderm layer, right? Then, sorry about that. Then the neural tube, hello? Hello, can you guys still see my screen? I'm sorry about that.
It's all right.
All right. You can confirm when my screen is up again.
Not yet.
What about now?
Still loading.
All right.
Yeah, it's now up.
All right. So I was saying that ectoderm is going to form structures that maintain contact with the external environment. Then we say that we had those three parts: surface ectoderm forming the epidermis of the skin. Then the neural tube is what is going to form basically the central nervous system, which is the spinal cord. You're going to look at the brainstem, the cerebellum, the cerebral cortices, and the diencephalon. Then I also want to talk about the neural crest cells. So you'd find in certain texts or books, they're going to be referred to as the fourth germ cell. And remember, I say that these cells are going to detach from the surface ectoderm when you have formation of the neural tube, right? So when they do that, they undergo an epithelial to mesenchymal transition, meaning they're going to change from an epithelial structure to a connective tissue structure, which is going to be mesenchyme, right? And when they are going to undergo this mesenchymal transition, they are going to migrate and they're going to reside in the underlying mesoderm. Remember, it's ectoderm, mesoderm, endoderm. So these cells are going to what? To migrate and reside in the underlying mesoderm. So these crystals, when they migrate in two ways. So they can either use a dorsal pathway or a ventral pathway. The dorsal pathway is the one way there is migration through the dermis, or basically they will migrate through the a part of skin, and they pass through the already formed basal lamina, and they form melanocytes in the skin. So crest cells that migrate through the dorsal pathway are the ones that form melanocytes for melanin in skin. Then the ones that migrate via the ventral pathway, they're the ones that use the anterior half of the somites, which we are going to find from paraxial mesoderm. And they form basically most of the structures that we're going to project on the next slide, which are coming from neural crest cells. That's sensory ganglia, sympathetic neurons, enteric neurons for the gut, and also the adrenal medulla. Remember, they say the adrenal medulla is like a displaced sympathetic ganglia, right? Then, so these are neural crest cells from the trunk. Then the neural crest cells, the cranial neural crest cells are the ones that we do when we talk about development of the head and neck. They're the ones that form craniofacial, the craniofacial skeleton. Then they also help in formation of glial cells and neurons for cranial ganglia for your cranial nerves, right? So I found this picture, which somehow tries to illustrate cranial neural crest cells in blue, truncal neural crest cells in green. Then there is sacral neural crest cells in, I think that's gray. Then vagal neural crest cells in pink, right? So notice for the truncal neural crest cells, they're the ones that we say those that take the dorsal pathway will form melanocytes. Then the rest for either the nervous system or the enteric nervous system, adrenal medulla, and the Schwann cells, those cells are going to be the ones that take the ventral what? The ventral pathway. Then cranial neural crest cells are the ones that form structures in the head and neck. For example, we say the craniofacial skeleton, and also odontoblasts when you do development of the teeth. They're also going to come from your cranial neural crest cells, right? Then sacral neural crest cells help with formation of enteric and sympathetic ganglia, right? Then for mesoderm, remember we said we're going to specify mesoderm into paraxial mesoderm, intermediate, and lateral plate mesoderm. Paraxial mesoderm is mostly going to be touched when we talk about development of the musculoskeletal system. But what I want you to know for now is, as an introduction, it forms segmented somites, which start to form in the third week of development. And these somites can help determine the age of that developing embryo, right? That somite will have a dermomyotome part and a sclerotom part. Demo part is dermis of the skin. Myotom part is skeletal muscle, basically striated muscle, right? Then the sclerotom part is going to form cartilage and bones of the vertebral column. Mind you, it won't include bones of the upper limb and the lower limb. That is also including the shoulder girdle and the pelvic girdle. Those bones of the limbs, they come from lateral plate mesoderm, right? Then intermediate mesoderm, for simplicity's sake, it basically forms genitourinary structures. So this is the layer of mesoderm that is mostly described when we do genitourinary structures. Then lateral plate mesoderm, for the person who do development of body cavities in the diaphragm, you find that it splits into two. And when it splits into two, it forms a layer closer to endoderm, which is going to be splanchnic, and a layer closer to ectoderm, which is going to be somatic. So for all the serous membranes, that's pleura, pericardium, or peritoneum, the somatic layer will form the parietal layer of pleura, of pericardium, and of peritoneum. Then the splanchnic layer closer to the endoderm or yolk sac will form the visceral layers, right? Then when we also do development of the heart, you find that the vascular system will also come from lateral plate mesoderm, right? So basically the vascular system is a mesodermal derivative, right? Then endoderm forms the lining of the inner structures, mostly the GIT and the respiratory system. So endoderm will form the GIT and the respiratory system. So when people say that, or when I'm saying endoderm will form the GIT, we are referring to the functional part of the GIT or the parenchyma within the GIT. So this is the glands and the epithelium. The connective tissue structures will then come from your mesoderm, right? Then other structures that are also going to come from endoderm will include glands, like your salivary glands, thyroid, the parathyroid. These, when we do the head and neck, you find that the parathyroid will be coming from your pharyngeal pouch, which you remember it's also going to be endoderm, right? Then the epithelial lining of the tympanic cavity and the auditory tube, which will be coming from your first pouch as well, are also going to be endodermal in what? In origin, right? So I've tried to make it a 40-minute presentation. I'm not so sure if we have questions before we move on to the discussion. Hello, are you there, guys?
Yes, we are there. No questions, I guess.
Okay.